Okay, so welcome to the Nearpod lesson that will accompany the visual system. So for these Nearpod lessons for Chapter 8, we're going to delve into the sensory systems in a little bit more detail than what we'll cover during the live lectures. So let's start with vision. So you probably already know this stuff from taking their intro, brain and behavior, some of the prerequisites for this course. But just quickly, sensory systems vision, this visual information processing begins in the retina. Of course, the retina contains those photoreceptors. So the receptors that detect light information. And we've got two types of photoreceptors. We've got rods and we've got cones. And of course, they serve slightly different purposes for vision, with rods being more concerned with detecting, motion and kind of those broad strokes of vision, of the visual experience. Cones being more concerned with fine detail, high acuity vision. So in the eye, the different structures of the eye, they work together to capture light and reflect it back onto these photoreceptors located at the back of the eyeball where the retina is. So, we've got the cornea and the lens. Cornea and the lens, these are what focus light, incoming light onto the back of the eye. We've got the pupil in here as well, where light actually enters the eye. But basically, all of the parts of the eye work to focus light onto the back of the eye where the retina is. So the retina is this receptive surface for light located at the back of the eyeball, containing those photoreceptors that detect the light. So really, the key thing that the retina does is it transduces that incoming light into neural signals. So we'll look at this in class, this kind of important concept of transduction, because really this is what the job of all the receptors, all of the different receptors that we have for our different sensory systems, this is what they all ultimately must do. They must transduce or convert whatever incoming stimuli we're talking about for whatever sensory system we're talking about into neural signals. So this is what the retina does. It transduces or it converts that incoming light information into neural signals. So just also quickly, the distribution of rods and cones across the retina, it's not the same. It's not uniform. So we have many more rods than we do cones, and they're also located at different regions of the retina. So in the center of the retina here, this is the fovea. So again, this should be review. The fovea, we've got a high density of cones in the foveal region. It's located in the center of the retina. So in the peripheral regions of the retina, off to the side of the fovea on either side of the retina, we've got a lot of rods. So many more rods in the peripheral regions than there are cones in the peripheral regions, and we have no rods in the fovea. So in the foveal region, this is our point of highest visual acuity. This is our point of fixation. So if you're really fixating on something and trying to see it really well with a lot of detail, you're going to be fixating your fovea on that location. So although we do have this point of fixation, the fovea, our eyes are continuously sampling different parts of the visual scene. So this means that all regions of the visual field can get continuously sampled by the rods and the cones. So our photoreceptors in our eyes, our rods and cones, they respond to movement of images across the retina. So if things are static on the retina, the receptors will stop responding. So movement of images across the retina is very important for activating rods and cones. So like I said, our eyes are continuously sampling different regions of the visual field, and this is done through quick involuntary eye movements called saccades. So saccades are quick involuntary eye movements, and these quick involuntary eye movements work to bring different parts of the visual scene onto the fovea. So even if we think we're holding our eyes still and we're trying to fixate on something, our eyes are not actually still. We're still making about 3 to 4 saccades per second. So these quick eye movements, they constantly shift the visual scene to different parts of the retina so that each part of the visual scene gets to take advantage of that high acuity foveal region. So this continuous sampling of the visual scene, this is what gives us, a clear image of the world. It doesn't appear to be clear at the center and blurry everywhere else. So these eye movements, they're so quick, we don't know we're making them, we don't know they're happening, but these saccades, these quick involuntary eye movements, they allow us to continuously sample the visual scene so that all regions of the visual scene can take advantage of that foveal region of high visual acuity. So in the retina, we have different types of cells besides just photoreceptors. So there's actually 5 different types of cells in the retina. So these 5 different types of cells, they have different functions. So let's just look at these quickly before we kind of talk briefly about what they do. So we've got our photoreceptors. These are these ones right here, our rods and our cones, which we've just looked at. We also have in the green, we've got horizontal cells. And next in the purple, we've got bipolar cells. And in the magenta, the pink, we've got amacrine cells, and then we've got ganglion cells in the blue. So this network of cells, a circuit of cells, transmits that incoming light information, which is converted to neural signals by the rods and cones. These converted signals travel through these different layers of the retina and ultimately end up in the ganglion cells, which forms the optic nerve. So the amacrine cells and the horizontal cells, they are similar in that they modulate the activity between the other layers that they are connected with. So if we're talking about the horizontal cells, they play a modulatory role. They modulate the activity coming in from the photoreceptors, from the rods and cones. They modulate the signals that are being sent onto the bipolar cells. So that's what is meant by a modulatory role. So they modulate, they influence the activity coming in from the photoreceptors, and they modulate those signals that ultimately get passed on to the bipolar cells. So the amacrine cells do a similar thing, but they modulate the activity between the bipolar cells and the ganglion cells. So they influence the signals that are being passed from the bipolar cells to the ganglion cells. So the amacrine cells and horizontal cells playing a modulatory role. Now, with the photoreceptors, they synapse onto bipolar cells, and the bipolar cells synapse onto the ganglion cells, but we do have these other two cell types kind of in between those connections that are influencing the activity that's passed between photoreceptors, bipolar cells, and ganglion cells. So once the signals are sent, so we've got our incoming light energy getting transduced by the rods and cones, getting passed onto the bipolar cells, and then passed onto the retinal ganglion cells. These retinal ganglion cells form the optic nerve, which is the beginning of the visual pathway that we'll be talking about in a minute. That's where the signals go from the eye to the thalamus and then on to the visual cortex. But first, a little kind of interesting side note about how incoming visual information lands on the eye. So the shape and curvature of the lens and the cornea, our eyeballs are curved, they're spherical, and the structures inside are also curved. So the shape of the lens and the cornea are really effective for bending light that enters the eye. And this is really effective for improving focus. Enabling those structures to really focus those signals onto the back of the eye. So you can see it kind of coming in like a bottleneck, right? The way that the lens and the cornea are able to focus that information with, this point here being, kind of the narrow end of the bottleneck. So really able to focus that incoming light information. And this is good, but what it means, the way that the light is bent, the incoming light is bent, it means that the image is projected onto the retina upside down and backwards. So if we're looking at this leaf, the way it is, the image is bent, it ends up being upside down and backwards. So that is what lands on our retina. An image that is upside down and backwards. But of course, this is not how we see the world, because our brain is able to correct this. So we have so much experience looking at the world, and through experience with how the world actually is, it's not upside down, our brains are able to kind of just disregard this reversal, and it actually writes the image for us. So we have experience viewing the world, and we know that it is not upside down. So our brains know that this is not the correct projection. So the brain is able to correct the final image that we see, writes the image so that we actually see a right side up image. So this is another, we'll kind of come across these examples throughout this chapter of how sensations may not always match our perceptions. And this is another example of how our perception does not always match the sensation, or we may not interpret it accurately. In this case, this misperception is quite useful. This right-siding, writing the visual image that lands on the retina. So let's look at the visual pathways from the eye to the brain. So let's first start looking at the optic nerve. So when I showed this image showing the distribution of cones and the distribution of rods, you may have noticed this little section here with the broken lines. What is that? Well, the axons of the retinal ganglion cells, they come together to form the optic nerve. But what this means is that the photoreceptors are pushed to the side where the ganglion cells, the axons of the ganglion cells leave the retina, leave the eye. The optic nerve pushes the photoreceptors out of the way, and this creates a blind spot. So I'm just going to flip back here for a minute because so one kind of key aspect of the flow of information through the retina is that incoming light information passes through all of these layers of cells, hits the photoreceptors, and then those signals are sent back out, and then the optic nerve, hitting the optic nerve, and this optic nerve comes back around and goes to the brain. So what this means for our blind spot is that where these axons come back around, it pushes the photoreceptors off to the side. And that means that there is a part on our retina that does not contain any photoreceptors. And that is what this right here is. It's called the blind spot. So where those axons of the retinal ganglion cells come together and form the optic nerve, this is where these axons exit the eye. They do it through the blind spot. So the blind spot is the place where the optic nerve leaves the eye, and there are no photoreceptors there. So let's do a demonstration of our blind spot. So let's do this demonstration to find your blind spot. It's really easy. So just look at the cross on the screen. So you're going to look at the cross on the screen and cover your left eye. So look at the cross, cover your left eye, and then I want you to move back and forth towards the screen and away from the screen very slowly. And you'll notice now you might have to experiment with different distances from your screen depending on how close or far away you are. But you'll notice as you move back and forth that there is a point where the dot disappears. So you just keep moving back and forth slowly until that dot disappears. And then you can just kind of keep doing it and see, yeah, that's very interesting, right? So that is your blind spot. So like I said, depending on the size of your screen, the blind spot may appear at different distances for everybody, but you just move slowly back and forth until the circle disappears, and this will be your blind spot. So we'll do a quick poll just to let me know whether or not you found your blind spot. So you can do that poll, and then we'll move on. So the visual pathways in the brain. So once the signals leave the eye through the optic nerve, they will cross over at the optic chiasm. So this is the optic chiasm, where we've got the optic nerves from each eye being combined. So at the optic chiasm, this is where input from both eyes crosses over. So let's just look at what this means in terms of the visual fields. So here we've got the right visual field and the left visual field. And we've got it mapped out in color, showing how it projects from each part of the eye onto the visual cortex at the back right here. So we've got a left visual field here, and we've got a right visual field here. So what you'll notice here is that both eyes are contributing something to each visual field. So we're not talking about left eye and right eye. We're talking about left visual field and right visual field. So in the visual fields, we've got some temporal retinal fibers. So these are located close to the sides of the head, the temples. You can think about the temple of your being on the side of your head. So we've got a left temporal region and we've got a right temporal region. And then we've also got a nasal region of the retina. And these are the ones, the parts of the retina that are closest to your nose. So for each visual field, we're getting information from each eye, basically. So when we're talking about the left visual field, that input is coming from the nasal region of the retina in the left eye and the temporal region of the retina in the right eye, and vice versa. So if we look at the right visual field, we are getting information from the right nasal region of the retina of the right eye. And we're also getting information from the left temporal region of the retina in the left eye. So what this means, we've got information from the right half of each retina projecting onto the right side of the brain. So the left visual field projecting onto the right side of the brain, right visual field projecting onto the left side of the brain. So, but we've got information from each eye contributing to the pathway that goes to each side of the brain. So when we're talking about this crossover point, the optic chiasm, we basically have about half of those optic fibers crossing over at the optic chiasm. So we've got information from the left half of each retina. This is perceiving the right visual field, projecting onto the left hemisphere, and vice versa. So this means that each hemisphere of the brain, the right and left hemisphere, is getting some information from both eyes. So after the optic chiasm, the signals are sent onto the lateral geniculate nucleus, which is the part of the thalamus that deals with visual information. So that's what the lateral geniculate nucleus is. It's the region of the thalamus that deals with visual information. As we'll see throughout this section on the organization of sensory systems, there's different regions of the thalamus that deal with the different senses. So when we're talking about vision, we're talking about the lateral geniculate nucleus. So the lateral geniculate nucleus, it has 6 layers. And this is an actual stain of what the LGN looks like. So you can clearly see those 6 layers, 6 layers of cells, clearly very distinctive. So the outer layers of the cells, outer layers of the LGN, they're called the parvocellular layers, or the P cells. So that's these ones right here. And the inner layers of the LGN are called the magnocellular layers. And these names simply come from the size of the cells. So the parvocellular layers, they are smaller cells. So parvo means small. So they are smaller cells, and the magno meaning larger. These are larger cells. So each of these groups of cells, so the parvo cells and parvocellular layers and magnocellular layers, they perform different functions. So the parvocellular layers are responsive to color, fine pattern details, stationary objects, so still objects, and slow moving objects. So the magnocellular layers, on the other hand, are more responsive to movement and light and dark contrast. So you can think about the parvocellular layers as being more concerned with those high visual acuity aspects of the visual scene, and the magnocellular layers being more concerned with the lower visual acuity aspects of the visual scene. So not surprisingly, the magnocellular layers and parvocellular layers do receive information differently from the rods and cones. So the magnocellular layers, they receive input from the cones, and the magnocellular layer receive input from the rods. Yeah, so parvocellular layers receiving input from cones, magnocellular layers receiving input from rods. Not surprising considering the different aspects of the visual scene that these layers are responsive to. So the LGN is divided into these 6 layers. And these 6 layers are important for understanding the topographic mapping from the retina to the LGN and then on to the visual cortex. So topographic mapping is just another way of saying that that the parts of the retina that are stimulated by stimuli from different parts of the visual field, this mapping is maintained throughout the visual pathway. So if a certain part of the retina is stimulated, that part of the retina is there's going to be kind of a one-to-one mapping between where it stimulates the retina, where it stimulates the lateral geniculate nucleus, and then where it stimulates the visual cortex. So this orderly mapping is maintained throughout the visual pathway. So we've got 6 layers of the lateral geniculate nucleus, and this mapping of the information as it lands on the retina, it's maintained in the lateral geniculate nucleus. So we've also got some different projections in terms of the eye that we're talking about. So ipsilateral, contralateral, these are terms that will come up a lot in this course, and probably terms that you should be familiar with. So ipsilateral meaning the same side of same side, contralateral meaning opposite side of the brain. So the layers of the lateral geniculate nucleus, they project differently. The layers project differently depending on the eye. So let's just look at this. So we've got our temporal and nasal retina coming back into play here. So we've got our right temporal retina, right nasal retina, meaning that it's coming from the right eye. So let's just look at how this projects here onto the lateral geniculate nucleus. So we've got our right and our left LGN. So, and this color coding I find is very helpful. So let's look at our right temporal retina, and where this information is going to the lateral geniculate nucleus. So it is going, so it's flowing along the optic nerve, it's hitting the optic chiasm, and then it's going to layers 2, 3, and 5 on the same side. So ipsilateral, meaning same side. So we've got the right temporal retina projecting onto layer 2, 3, and 5 on the same side. So then we've got our right nasal retina, and it's doing something a little bit different. It is projecting onto layers 1, 4, and 6 of the other lateral geniculate nucleus. So the left lateral geniculate nucleus. And then the same thing happens for the left eye. So we've got the left, the nasal retina of the left eye, so the one closest to the nose. It's projecting onto the contralateral side, meaning that it's going to be hitting layer 1, 5, and 6 of the eye of the lateral geniculate nucleus on the opposite side, and then the remaining temporal regions in the left temporal retina going to be projecting to the same side, the left lateral geniculate nucleus. So the lateral geniculate nucleus then projects to the primary visual cortex, also called V1. And it projects very specifically to layer 4 of the primary visual cortex. And again, these projections keep their organization. So if we look at our layers again, so our layers 1, 2, 3, 4, 5, 6. So we saw in the previous slide how the different regions of the visual field project in a very organized fashion onto the left and right lateral geniculate nucleus. And then this organization is maintained as it projects onto the visual cortex. So let's look at this. So we've got input, basically input from the left eye alternating with input from the right eye. And this is what gives the visual cortex a striped appearance when it is stained. And this is why it's sometimes called the striate cortex as well. So primary visual cortex, V1, striate cortex, all means the same thing. So let's just look, let's look at this. Okay, so we've got our optic tract coming in, we've got our lateral geniculate nucleus. So let's just look at the different layers. So these are our magnocellular layers here, 1 and 2. So they are projecting, so if we look at this section here, this one section is receiving one layer of magnocellular pathway, and then the next section of the visual cortex is receiving this input from the other layer. And then if we look at 3, it's going here, 4, going here, so and so on and so forth. So we've got kind of this alternating input between each eye coming in, and these additional color here, the yellow and mauve, indicating either contralateral or ipsilateral input from each eye. So you can see that this organization, this topographic mapping, topographic organization is maintained from retina to lateral geniculate nucleus right through to the primary visual cortex. So this pathway is called the geniculostriate pathway because it's projecting from the lateral geniculate nucleus onto the striate cortex, primary visual cortex, V1, got a few different names, but it all means the same thing. So from here, we've got two other pathways in the brain where these signals start to diverge after they leave the primary visual cortex. So from the visual cortex, these signals, they diverge along two processing streams. They are called the dorsal stream and the ventral stream. So the dorsal stream, also sometimes referred to as the where stream or the action stream, because it's involved in using visual input to perceive the location of objects and help us to guide our movement towards them. The other pathway that we have projects from the primary visual cortex along the inferior temporal lobe. It's called the ventral stream. And this is sometimes called the what stream because it is more involved with visual identification of objects or object recognition. So we do have another pathway. So we had the geniculostriate pathway. We've got this other pathway. It's called the tectopulvinar pathway. So this pathway, it projects, so if we look here at this image, so this is what we were just talking about. So retina, optic chiasm, back to the primary visual cortex. This is the pathway we were talking about. So just a minute ago. So this other pathway that we have, instead of projecting to the visual cortex, it projects to the superior colliculus, which is right here. So some parts, some fibers, when they leave the lateral geniculate nucleus, most of them project to the visual cortex, but some of them project to the superior colliculus. And this is what makes up that tectopulvinar pathway. So it is, sorry here, I'm getting ahead of myself. So projecting before it gets to the thalamus, projecting to the superior colliculus. So coming off of the, branching off of the optic nerve. So this pathway is very fast acting, and it is important for providing us with information about kind of absolute spatial information of objects. And it's very important for orienting towards visual stimuli. And so if you think about a flash of something, seeing a flash of something out of the corner of your eye, you immediately orient towards the flash, the flash out of the corner of your eye. So that is the tectopulvinar pathway at work. So, when you see that flash out of the corner of your eye, you don't know what it is until you orient and you can see what it is. So this pathway is important for orienting towards visual stimuli. So this pathway is, very important for, visual orienting, quickly locating objects in space, but it doesn't tell us what the object is. So it doesn't provide detailed information about what we're looking at. So ultimately, these pathways are going to project to the visual areas in the parietal lobe and the temporal lobe and what have you. So eventually, and by the time these pathways reach, the parietal lobe or the temporal lobe, the rest of the visual system has already had time to catch up, and we would know what we're looking at, what we're orienting towards or away from, depending on the situation. So initially we don't know what we're looking at, but by the time we orient and have looked, the other pathways have had time to kind of catch up and identify what we're looking at. So this brings us to an interesting kind of clinical condition known as cortical blindness. So cortical blindness is a condition that can happen as a result of damage to different regions of the visual pathway. So cortical blindness, this is a loss of vision that is not caused by problems with the eyes. So if a person has cortical blindness, their eyes work fine, but they have damage to different regions of the visual pathway. So cortical blindness, there's an interesting condition that's related to this, which is called blind sight. And this is when people who have cortical blindness as a result of damage to the visual pathway are still conscious of stimuli in their environment. So loss of vision they experience, but they may still be conscious of stimuli in their environment. So we'll watch an interesting video on this in a minute, but let's just look at what this can mean. So cortical blindness, so you can have small lesions in the visual cortex or other parts of the visual pathway as well. And these small lesions, they can be small, they can be large. And the size of the kind of what is referred to as a scotoma will vary depending on the size of the lesion. So if you have small lesions in the visual cortex, it can produce something called scotomas, which are small blind spots in the visual field. So they may appear as dark spots. So if you look right here in this, on the slide, this is an example of that. So they could be dark spots, they could be light spots, they could be blurry spots. So basically you have some parts of the visual field that are obscured in some way. So often, you know, depending on the size of the scotoma, they may not cause the person any problems, you know, because, well, for one, we've got two eyes that are constantly moving. So our brain is able to fill in this missing information, you know, depending on the size of the scotoma. So there are some research or experimental techniques that can be used to essentially trick the visual system into revealing these scotomas. So basically, you can trick the visual system into revealing the scotoma by placing objects entirely within the region of the scotoma. So placing objects entirely within the scotoma region of the person's visual field, and doing so without allowing the person to shift their gaze. And then you ask them what the object is. So if they don't see an object, the experimenter, the researcher, you move the object out of the scotoma so that it suddenly appears in that intact region of the visual field. So this is what kind of a trick that can be used to demonstrate, you know, the scotoma or kind of the blind region of the person's visual field. I mean, and you can kind of think about it as being similar to the demonstration that we just did, showing our own blind spot. So basically that's what would happen. Instead of, you know, the dot appearing or disappearing, it's the same type of concept, right? You move the object into the person's scotoma, it disappears, you move it out, it appears. So let's look at a case study of this. The case of blind sight in a person with cortical blindness. So DB, a very famously studied person with blind sight. So he has a large lesion in his occipital cortex. Basically, he had his right calcarine fissure removed. So the calcarine fissure, basically just another anatomical name for visual cortex. So he had majority of his right visual cortex removed as a result of an angioma. So if we're looking here, this is a CT scan showing this angioma, which is, it's basically all it is a collection of abnormal blood vessels. So that's what this is showing right here. Collection of abnormal blood vessels. And this causes disruptions to blood, disruptions in blood flow or abnormal blood flow to different regions of the brain. So because of this angioma, DB had his right calcarine fissure removed to get rid of that angioma. So then he was losing a significant part of his visual cortex. So basically what he has is a scotoma that encompasses his entire left visual field. So right side, right calcarine fissure removed, basically the entire left visual field is not seen to him. So he would report seeing nothing when it's presented in his left visual field. So he'll report seeing nothing, but he will also report that he has a feeling that a stimulus was approaching. And he may even describe it as being a smooth stimulus or a jagged stimulus. But he would still insist that he saw nothing and figures that he was kind of just guessing at whatever he felt like he was seeing. But if DB was tested, so if he was asked to kind of point to locations in his impaired visual field where, say, spots of light were being briefly flashed, he was surprisingly accurate. So he would report seeing nothing, but if these spots of light were flashed in his defective visual field, he could report where they were with surprising accuracy. So this really contrasts with his subjective experience of seeing nothing at all, but still being able to report with quite good accuracy where the location of these flashes of light were. So basically, with DB, he has cortical blindness, complete loss of a visual field, where he reports no conscious awareness of seeing, but he is still able to report the movement and locations of objects, even though he can't recognize them. So we will look at a brief explanation for this really weird phenomena of blind sight, which is basically best summed up as unconscious visual processing. So where the person is not consciously aware of objects in their defective visual field, but yet they can still detect them with quite good accuracy. So what are some potential explanations for this? One is that some visual abilities are maintained through intact areas of the visual cortex, which may be perhaps taking over for those damaged areas. But another explanation relates to that technopulvinar pathway that we were just talking about. So this is a subcortical pathway, the technopulvinar pathway to the superior colliculus. And this pathway may be maintaining some visual abilities. So we talked about the primary visual pathway from the lateral geniculate nucleus to the visual cortex, but this is that other one, the subcortical one that runs through the superior colliculus. So we've got about 10% of the axons emerging from the retina and projecting to the superior colliculus through this technopulvinar pathway. So this pathway, it's considered a primitive pathway that's involved in these quick orienting responses that kind of need to be so quick that they don't pass through the cortex, right? So they don't pass through the cortex and allow you to know what the object is. And you can imagine why this would be important as a primitive pathway. So if something comes flying at your head, you may instinctively duck without first knowing what is actually flying at your head. So this is the result of this subcortical tectopulvinar pathway to the superior colliculus. So it doesn't matter if you can see what's coming at your head. It matters that you duck first and then figure out what it is later. So in terms of blind sight, people with this condition may be aware of something being there because of this subcortical pathway to the superior colliculus. And through this pathway, they maintain some rudimentary visual abilities, but they do so in the absence of any awareness. So this would explain why they have a feeling that something is there, but they don't know what it is. Okay, so let's watch a video from Dr. Ramachandran illustrating a case of blind sight.